Okay, guys, I'm sorry for being late. I don't know, the rest of them might be attending online or whatever, but you are ready, Kalisha, right? So let's invite Kalisha Lal for this FPS. So Kalisha Lal, you will give a small introduction about yourself, and then you will get on with your presentation. Okay, let's go.
Good evening, one and all. First of all, I would like to thank director to give me the wonderful opportunities present this science forum. Thank you, sir. I want to introduce myself. I'm Kali Solal. I did my PhD in Kalasaling University, India. After that, I have been completed 4 years postdoctoral study in various countries. So today I'm going to, I'm glad to share my research experience with you all. I hope everyone enjoy my talk. I have shown a few topics which I have going to focus in my today topic. Let's see one by one. Selenium nanoparticles role in cardiac tissue engineering. Before going to this topic, the small introduction about what is selenium and selenium deficiency. Everyone know about selenium. Selenium is an essential element in our human body and acts as an antioxidative property in cardiovascular disease. What is selenium deficiency means if the low level of blood plasma selenium less than 60 microgram per liter, that is called selenium deficiency. In all over the world, the selenium deficiency was affected 0.5 to 1 billion people due to the food intake of selenium differ in various parts of the world. Mainly agriculture soil containing low level of selenium which affect our human health. There is a different concentration of selenium present in our human blood. One is deficiency, second one is optimal level of selenium, another one is excessive. As I said yearly, if the low level of blood plasma selenium, which affect increasing risk of cardiovascular disease, viral virulent, prostate cancer, etc. Similarly, the high amount of selenium which affect a lot of complications such as skin cancer, prostate cancer, type 2 diabetes, etc. The optimum level of selenium 125 microgram, which is more safe from free radicals and cancer risk. So as per the WHO report, the daily intake of selenium male around 75 microgram per day, in female 60 microgram per day. This is the overview of selenium present in our human blood. So when I did my PhD, this paper came from New England Journal of Medicine. This is one of the prestigious journal in the medicinal field. They published a mechanism of selenium deficiency to cardiomyopathy. As I said earlier, if the low level of blood plasma selenium, which is decreasing glutathione peroxidase, which increasing oxidative, further leads to increasing oxidative stress, finally cause cardiomyopathy. Based on this paper, why not we develop selenium nanoparticle as a cardiac tissue engineering purpose? These are my PhD thesis. So let's move on to selenoprotein. Selenium, I'm more interested in selenoprotein. Selenoprotein is a protein that include selenocysteine amino acid present in the each sequence of selenoprotein. There are a lot of selenoprotein available in our human body. There are some of the protein functionalized, some of these not yet functionalized. So here glutathione peroxidase, thyroidector reductase, most of the function characterized, which is only one selenocysteine present in the amino acid. As you can see, this structure of the cysteine and selenocysteine, There is only difference is sulfur group replaced to selenium, which is involved in the translation process. In the translation process, there is a more complication process. I'm not going to discuss about today here. And another recently, in 2012, Stockwell Group published GPX4 is one of the main regulator for ferroptosis pathway. Ferroptosis is nothing but is ion-dependent pathway which regulate in the lipid peroxidation. So it's a GPX4 main regulator for this ferroptosis pathway. In over the last few decades, drug discovery, more than 75% of protein research still focus on the 10% of protein. Even though many protein available in our human body, for example, selenoprotein has been genetically linked with disease, particularly cardiovascular disease and cancer risk. So in my future study, I'm going to focus on selenoprotein related with cancer research. Then now we are focusing on detail of selenoprotein. In this schematic diagram, hope everyone understand. GPX1 is one of the selenoprotein which need a selenium from the diet. In the GPX1 protein containing selenium that include the selenium. It is a function of particular function in the GPX1. When the free radical come alone, it can be bind with the GPX1. Selenium is a major role involved in the catalytic property of a particular enzyme. Finally, the free radical broken into two pieces with the help of seleno enzyme. Before going to any damage occur in the free radical, the selenium, because of selenium enzyme to protect the free radical from the sample. As I said previously, the New England Journal of Medicine published one hypothesis, selenium deficiency, which decreasing the glutathione peroxidase. Similarly, if more amount of selenium, which involve to GPX1 activity to protect the cardiovascular health modulator. So whether this hypothesis is true or not, we studied epidemiological parameter between selenium and human cardiovascular disease in Tamil Nadu population. First, we collaborated with Madurai Meenakshi Mission Hospital, and then we got human ethical clearance from this hospital. After that, we collected 60 patient different area of Tamil Nadu were studied. Among 30 controlled, 30 cardiovascular patient. With the help of doctor, we collected sample from blood sample from the both group, and then we analysis so many parameter like selenium concentration measurement using ICP-MS method, and HRP sensitive marker for cardiovascular disease using nephrometric assay, and cholesterol measurement. All the parameter we did, we give the commercial lab. They provide some result from the sample. In this table, there is no significant difference were observed in the lipid profile, but how to differentiate control and cardiac patient means we analysis HRP, high sensitive C-reactive protein, one of the sensitive marker for cardiac tissue damage and inflammation in the sample. So based in the HRP protein was more elevated in cardiac patient when compared to control. Based on the HRP protein, we differentiate two different group, and further we analysis selenium and glutathione peroxidase assay. In this graph, clearly shows that there is a significant difference were observed both control and cardiac patient. The above epidemiological parameter conclude that selenium supplement could improve the cardiac health modulator. So next we move on to nanotechnology field. Nanotechnology can able to synthesize nanomaterial. which produce the electrical property and the mechanical property. That's why we develop nanoparticles from bovine serum albumin keratin molecule. Let's see the mechanism of cellular nanoparticle synthesis. When the protein was exposed to heat, the structure of the bovine serum albumin will facilitate and breaking into unfold structure of the protein. In this unfold structure of the protein was exposed to more number of SH group. This SH group could reduce the reduction of selenium into cellular nanoparticles, which is confirmed from the based on the color change reaction. We confirm that as you can see the photograph A, BSA alone and BSA plus sodium selenate change red color, which is confirmed from cellular nanoparticle present in the sample. But interesting study note that when the same reaction was carried out with the inhibitor study N-ethyl malamate, it's a specific inhibitor for SH group, they could not produce any color change in the reaction solution. Based on this inhibitor study, we conclude that SH group is a main role involved in the reduction of selenium into cellular nanoparticles. The successful synthesis of cellular nanoparticle was characterized to so many techniques. One of the technique called scanning electron microscope. In this result, we shows that morphology characterization and size of the nanoparticle were observed, which is confirmed from EDAX mapping and EDAX spectra. The EDAX mapping, you can see the red color spot indicate the presence of selenium element and the green color spot presence of sulfur element. And further we characterized in the EDAX spectra also. And then another one characterization study, dynamic light scattering is one of the particle size distribution assay. The BSA mediated cellular nanoparticle synthesis around 353 nanometer size. The keratin mediated synthesis around 145 nanometer size. Then whether these nanoparticles are stable in water or not, then we check zeta potential.
Kalishwaran, let me ask this question. So all those SH groups that you showed, they are the intrinsic cysteines of BSA, right?
Yes, sir.
So BSA, how many cysteines do BSA have?
Around 120, I
120. So all these 120 cysteines will occupy selenium.
think. Yeah.
So that entire thing, you call it as a nanoparticle, or this will break into small pieces?
Small pieces, the main role is SH group when the heat when the heat provided selenium disulfur bond break into the small piece, and then SH group is be exposed on the unfold structure of the protein.
Yeah.
Then this unfold structure of the protein SH group main role to reduction of selenium into cellular particles.
True, but still it's one single protein chain, the polypeptide chain, right?
Yeah.
So the BSA opened up. All the SH groups are exposed, and that will occupy one selenium at each of the 16 positions.
Yes.
But still it's one single big molecule, right?
Yeah.
So why do you call it a nanoparticle?
So it's formed the nanoparticle which is confirmed from EDAX mapping. As you can see, this is only red color spot indicate the selenium only.
Yeah. That's why I'm asking. So it is the entire BSA chain staying as it is.
So after that we centrifuge, then we collect the sample from supernatant.
So do you cut this BSA chain into smaller pieces by any chance? No.
No.
Do you sonicate it?
No.
And no enzyme treatment also. So each particle will have one protein molecule, or like that's
maybe we don't know, sir.
Okay. All right.
So based on the characterization study, we synthesized two different nanoparticles. One is 400 to 500 nanoparticles using BSA. Another 100 to 200 nanoparticles from keratin mediated synthesis. So cellular nanoparticle could be used as a therapeutical purpose. Before going to therapeutical option, we are going to study toxicity study in zebrafish embryo. Why you prefer zebrafish embryo as a toxicity effect? Zebrafish and human, it's a more 85% homology genome match with the zebrafish genome. So this embryo is easy to breeding and maintain in the lab, it is more transparent, so easy to watch heartbeat and development. Now, in another advantage of zebrafish embryo is so no need to dissection. You can see the assets, the toxicity could be as well as asset. So because of this advantage, zebrafish were selected as an experimental model for our study. So first we exposed to 5 different concentration of selenium nanoparticle treated. As you can see this photograph, A is a control, and B to F is a different concentration of selenium nanoparticle treated. This is the outer surface of the chorion. The concentration dependent selenium nanoparticle observed in the outer surface of the chorion. As you can see this, the black color spot indicate the selenium nanoparticle present in the outer surface of the chorion. After that, we analysis mortality, viability of a zebrafish embryo were studied at low concentration of selenium nanoparticle treatment. There is no mortality were noted. At the higher concentration of selenium nanoparticle treatment, there is a huge number of survival death occurred when compared to the control. So the embryo enter into the larvae after 24 hours. Embryo enter into the larvae stage. In this stage, we observed abnormality of zebrafish larvae stage. As you can see this photograph, the low concentration of selenium nanoparticle did not show any malformation such as pericardial edema and tail malformation. In the case of high concentration of selenium nanoparticle treatment, there is we observed tail malformation, short tail and curved tail we observed, particularly in the pericardial edema in the heart region. Next slide, I show the clearly in the circle color indicate this is a cardiac region of zebrafish larvae stage. When compared to control, the huge inflammation were observed in the higher concentration of treatment. Similarly, we observed in the videograph heart rate development we observed. There is a decreasing heart rate were observed in the 25 microgram selenium nanoparticle treatment when compared to control. So based on this toxicity study, helped to identify the toxic concentration to find out the therapeutic concentration for my further study. So we prefer 5 to 10 microgram could be used as a therapeutic concentration for cardiac tissue engineering purpose. So in 2012, another one paper came from a Nature group. They suggest that selenium, it's a reprogramming of fibroblast differentiate to cardiomyopathy. So these are more supporting my research. So we develop a novel biocompatibility chitosan selenium nanoparticle film with electrical conductivity and as well as mechanical property for the cardiac tissue engineering purpose. First, we develop this film, and then we seeded H9C2 cell line in the top of the film. After 24 hours, cells are growing with which analyzes cell viability assay and scanning electron microscope study. So if you want to detail, please refer this paper. I'm not going to discuss further. Then after finishing my PhD, I went to postdoctoral study in Ariel University, Israel. Here we develop a gold nanoparticle sustained peptide conjugation for the targeted drug delivery to cancer cells. Before going to results, a small introduction about this topic. Everyone know about cancer drug has more cytotoxicity effect in both the cancer cells and normal cells. So our aim is to enhance the therapeutic efficacy and reducing toxicity in the particular non-targeted tissue. So this is the main aim of our study. So we develop a peptide drug conjugation as a development of to targeted delivery to the specific cancer cell. But one of the main challenge of this PDC is not stable in the blood, liver, kidney. So to overcome this problem, we develop alternative strategy called PEG coated AU nanoparticles could be used as the could be suitable platform for the bioavailability of PDC and cytotoxicity effect through the particular targeted cancer cell line. So we prefer a Phage display peptide library, one of the most common searching of peptide library. This technique is a powerful tool to discover the novel peptide against any targeted antigen or cancer cell line. So we brought a Phage library from New England Journal of Commercial Lab. Then this is a M13 E. coli Phage. The P8 is a head region, the P3 is a tail region, and that this trail region, there are so many Phage will be cloned. That is called a Phage display library. And the random display in the particular peptide in this region, the Phage library we expose to particular target affinity, for example, antigen or cancer cells. After that, we remove the unbound Phage from the targeted site. Then we choose a bound Phage in the further analysis for characterization and peptide characterization based on this bound Phage. So these are the technique we develop in my lab. This is a very unique method compared to previous biopanning method. First, we collect the Phage library from the commercial lab. Then we injected into the mice. Then we collected unbound Phage from the mice, and again we infected with the particular cancer cell. For example, chronic lymphatic leukemia is one of the cancer we studied, our targeted cancer cell line. And then we prefer to collect the internalized Phage. Why we are preferring internalized Phage means some of the bound Phage is not stable, so that's why we prefer to internalized Phage and further characterization for DNA purification peptide sequencing. So we selected three different peptide, P4, P6, and P8 with the conjugated FITC. We exposing A20 cell line in a concentration dependent manner. Again, the P4 is a more high affinity in the concentration dependent manner when compared to other peptide. So P4 is a main target for particular cancer cell line. So again, we studied flow cytometer analysis in off-targeted cell line. The PBMC is a normal cancer normal cell line. The MOC is a NP4 HL60. Those cell lines are acquired leukemia cancer cell line. So one of the blood cancer cell line. We analyze all the cell line, but interesting study, we note that P4 peptide only bind with the A20 cell line. Again, we synthesize peptide drug conjugation in the click chemistry, and we synthesize three different peptide, chlorambucil, melphalan, pentamycin. In this figure, we shows that stability of PDCs were observed. In this graph, there is a T half only 20 minutes it will be degraded, which is confirmed from LCMS method. So in our alternative strategy called cold nanoparticle PDC, which is more stability up to 24 hours, which is confirmed from LCMS method. Finally, we exposed to toxicity study in the particular cancer cell line and off-targeted cell line. First, we effect of PDC, the pre-incubation with media, and then we exposed to A20 cell line. After 24 hours, which is growing growth curve were measured using XTT assay. There is no cytotoxicity were observed in the other cell line, human HL60 NP4 MOP cell line. So finally, we conclude that cold nanoparticle PDC is more targeted to particular A20 cell line. It is a slow release formulation of targeted to cancer cells. After finishing my first postdoc, then move on to second postdoc where I did my postdoc at University of Pittsburgh, USA. Here we studied the molecular mechanism between ferroptosis and apoptosis. everyone know about apoptosis cell death pathway. There is two pathway. One is death receptor signaling pathway. Another one is mitochondrial signaling pathway, which is lead to caspase activation, finally cause apoptosis. But another one cell death pathway, pharoptosis, this is a ion-dependent accumulation of lipid peroxidation rather than caspase activation. So in this two pathway, to investigate the possible mechanism, which is the main regulator for the particular pathway, we are going to see this pathway. So we prefer apoptotic agent as a trial, pharoptotic agent as a erastine molecules for my study. First, we check whether this drug alone, erastine alone, there is no cytotoxicity were observed. But trial alone, there is no cytotoxicity were observed. But the interesting study note that in the combination of two treatment, there is huge amount of cell death were observed, which is confirmed from the apoptosis marker. You can see the PAR cleavage is one of the apoptotic marker, which finally a downstream molecule of caspase 893 is activated, finally cause apoptosis. So whether this is a caspase mediated, we check inhibitor study. This is a cleaved caspase inhibitor study with a combination of two treatment. There is no cleaved caspase 3 were noted to protect the combination of two treatment. So based on this study, this pathway is mainly regulated to intrinsic pathway. So mitochondria is a major role involved in this pathway. So we did a knockout study from BAX. BAX is one of the pro-apoptotic molecule to regulate the mitochondrial signal pathway. So in this knockout study, there is no significant PAR cleavage were observed in the BAX knockout study when compared to BAX knockout study. So finally, PAX is a main regulator for involving in this combination of two treatment. So again, we collected the mitochondrial fraction was isolated, and then we conjugated with the FAX antibody. Then we run the sample. There is a multimer formation of when the combination of treatment, this multimer were formed when compared to other single treatment. So finally, the BAX is oligomerization to disturb the mitochondrial membrane, which is confirmed from confocal analysis. This JC1 stain mitochondrial confirm the loss of mitochondrial membrane, and then the cytochrome release from the mitochondria and to involve in the activation of caspase 3. So finally, we conclude the combination of two treatment to enhance the apoptotic mainly activity of caspase 3 through BAX associated mitochondrial dependent intrinsic pathway. So the above research, here I am going to establish my future study to understand the metabolic selenium heme trace element at the molecular level with the crosstalk between other pathway. We are going to discover the new cancer drug from this pathway. Another one study we are going to establish in our lab, exosome-mediated cold nanocrystal synthesis and its application in bioimaging purpose. Finally, this is to evaluate the pharmacological dynamic of Erythrocyte B against hepatocellular carcinoma. So it's a time to acknowledge my mentors. I would like to thank my mentors who helped me the entire my PhD postdoctoral life. My special thank to Dr. Ruby ma'am to give me wonderful opportunities for MK Band Fellow. Finally, I thank funding agency TBT. Thank you, Ananda.
So anybody has any question for Kalishwar Lal? Yeah. The mic is coming. Hello.
Yeah.
Sir, I have one question. So when you use the nanoparticle and you are checking their cardiac impairment, so what do you use as control for nanoparticle treatment?
So control, we did sodium selenate as a control. I'm not showing here, but
So sodium selenate at the same concentration you're using?
yeah, same concentration.
Okay. And how are you connecting the nanoparticle with apoptosis story? So is there any connection between this two?
No, this is a very, this is a separate chapter. The first chapter I did my PhD. The second chapter where I did my postdoctoral study.
Okay, so actually I was trying to find out whether you are connecting those two stories. So, anyway, so that's, thank you.
His point was anytime like you talked about selenium or nanoparticles, then you talked about one of the nanoparticles. I forgot what it was. What was the second nanoparticle that you created?
Gold nanoparticles.
Gold nanoparticle. Then you are now talking about ferroptosis.
Ferrop.
So is there any plan for you to connect your studies together anytime in future?
In future, I will study the overall selenium and heme as one of the main regulator to crosstalk between other pathway.
So this selenium nanoparticle that you made, is
Yeah.
anybody using it now for any purpose? Because you did your PhD,
Yes.
then you left and you did two postdocs.
Yeah.
So that nanoparticle must be lying there.
Yeah. My previous boss did in vivo study.
Okay, so what's the thing like? What's the story? Is there anything coming out of that?
I don't know. I didn't discuss about that.
Okay. Yeah, guys. Anybody else has any question? Yeah, seems nobody else has any question. But in case you have any questions, see, he can make nanoparticles. So you can talk to him about nanoparticle making in case anytime you are interested in making nanoparticles with anything. So thank you,
Thank you.
Kalishwaralal.
Thank you.
Thank you guys for coming.